Hello everyone, we are the East Coast Bio Crew. Today, we are here to introduce you all to our project, RhizeUP, and how genetically engineered rhizobium can become the future of combating nutrient runoff and protecting our waterways. Red tides. Dead zones. These are two devastating effects of eutrophication and overabundance of nutrients in waterways. Often, these increased nutrient levels are a result of runoff carrying excess fertilizer from farms. Only 20% of the phosphate in fertilizer is actually used by plants, leaving the rest to be washed into lakes and rivers where they fuel the growth of harmful algae. When algae grow, they use up oxygen and nutrients in the water, depriving aquatic life and animals in the process. Scenes like this are becoming ever more common since phosphate fertilizer use is on the rise. It is estimated that worldwide consumption of phosphate fertilizer has reached over 20 million tons annually. The results are that algal cleanup and prevention effort in the U.S. alone have cost communities over $1 billion over the last 10 years. Even though the struggle to contain nutrient pollution is global, the demand for fertilizer still grows. While prices are stabilizing from the previous year, the combination of the COVID-19 pandemic and the war in Ukraine has emphasized the potential for fertilizer prices to spiral out of control. Russia accounts for 12% of the world's phosphate fertilizer trade, according to the Western producer, and 25% of the world's nitrogen fertilizer trade, according to the NPR. In these uncertain times, we especially see a need to look into different innovative means of providing nutrients to our crops. We are the East Coast Bio Crew, a group of high school students from different schools who work at community labs based in Baltimore and New York City. The primary objective of our project RhizeUP is to reduce the threat of algal blooms on our waterways. To achieve this, we chose to genetically engineer Rhizobium, a soil bacterium known for its symbiosis with legumes, to have the capacity to solubilize and uptake mineral phosphate to reduce the use and reliance on phosphate fertilizers. When it comes to implementing our project in the real world, we have planned a multifaceted approach involving collaboration with stakeholders like farmers, private companies, and landowners, as well as organizations such as the Chesapeake Bay Foundation and government extensions like the Environmental Protection Agency and the United States Department of Agriculture. In farms, our project can be applied to legume crops in order to increase phosphate availability. We also plan to create phosphate sponges with inoculated plants in bank regions where runoff is known to be an issue. This is where the solubilization will really shine, and how we will complete our goal of reducing the use of fertilizers. As the engineered rhizobia solubilize and uptake the inorganic phosphates, the now phosphate-rich organic matter left behind by the plants can be composted and reused as a new biofertilizer. In terms of use by the government and other organizations, we hope to establish our project at the edges of the Chesapeake Bay and Hudson River in order to act as filters for phosphate runoff. In the long term, we aim for our project to promote sustainable practices in the agricultural industry with a novel regenerative agricultural paradigm. This will be a massive shift for the agricultural industry as a whole. Our application of synthetic biology is a proof of concept for the recycling of phosphate from fertilizer, a resource which until now could be considered a non-renewable resource. Again, this is especially important considering the wide-reaching effects of geopolitical conflict on commercial fertilizer markets, as we've previously mentioned. Our plan is to have a two-pronged approach, to uptake more mineral phosphate and to solubilize mineral phosphate. To increase the uptake of mineral phosphate, we are attempting to add new functions to our organism by inserting the pstSCAB operon, an organic phosphate transporter from E. coli. This operon contains genes for the pst, an inorganic phosphate binding protein that delivers phosphate to the membrane channel in the periplasm of a bacteria formed by pstA and pstC. pstB is an ATP hydrolyzing protein that energizes the phosphate transport into the cell for the pstABC transporter. By increasing phosphate uptake, we hope to reduce its presence in our environment. To solubilize mineral phosphate, we are attempting to introduce the secretion of gluconic acid by the PQQC plus GCD pathway from Agrobacterium radiobacter. Here, the substrate glucose is enzymatically converted to gluconic acid by the enzyme glucose dehydrogenase, which is coded by the GCD gene, and helped by the redox cofactor pyrroloquinoline quinone, coded partly by the PQQC gene. By solubilizing mineral phosphate, we hope that it increases phosphate availability for plants and eliminates the need for excessive phosphate fertilizers.
When we started working with Rhizobium, we needed to determine the best growth conditions since we saw several different growth media used by previous studies. On the math modeling team, we made models based on optical density measurements taken in our lab. They also follow the assumption that the bacterial growth follows a sigmoid curve. The general equation for that is shown here, where A influences the maximum growth rate, B is the time at which the growth stops being exponential, and C is the maximum optical density. The first test we conducted was to determine the optimal growth medium for growing Rhizobium in the lab. We compared ATC C111, a soil-based medium, against yeast mannitol extract with either high or low calcium. We determined that the optimal medium among those was yeast mannitol extract with low calcium because it provided the best growth rates and accuracy due to the low turbidity. Once we started working with plants, we were unable to see our Rhizobium form root nodules. While troubleshooting, we realized the temperature could be a reason, so we tested to see if Rhizobium needed a specific temperature to grow optimally. However, there was very little improvement in the growth rate at the optimal temperature of 30 degrees Celsius compared to the room temperature that we were using to grow our plants. So while we moved some of the plants to a warm incubator, we think that some of the factors may be responsible for the lack of nodulation. Finally, we found that the Yale 2016 team used tryptic soy broth to grow the Rhizobium. And when we compared growth rates in that medium to growth rates in our yeast mannitol extract, low calcium medium, we found that it was much more effective media. This discovery occurred too late to influence our project. If we were to continue with this project in the future, we should use that medium instead. Because we had difficulty finding information about working with Rhizobium when we started, we put all our findings together into a fact sheet for future iGEM teams. Next, we worked on cloning our genes. After a successful blue-white screening to confirm the integration of the gene in our plasmid, we transferred the plasmid from E. coli to Rhizobium and screened our Rhizobium by PCR. Our PCR and gel electrophoresis experiments showed that our transference of Rhizobia was successful. From the image below, several of our samples show successful gene insertion of the pstSCAB genes, and several also show successful gene insertion of PQQC plus GCD genes. Lastly, we wanted to confirm whether our genes could successfully synthesize our needed proteins. We ran a protein gel on Rhizobium carrying the genes and saw promising results from GCD protein synthesis. We expected 87 kilodaltons for the GDH enzyme and saw a corresponding band for one of our PQQC plus GCD samples. The arrow marks the band in the image below. It is worth noting that we were expecting to see the proteins running at their individual sizes, but because pstA, pstB, and pstC form the pstABC transporter and therefore form strong associations, we were also aware that we might see them still associating in the gel and a protein complex at a larger size. This would correspond to around 95 kilodaltons. To determine whether inserting the pstSCAB genes increased phosphate uptake from the media, we looked at our engineering bacteria's capability to uptake phosphate from plates with high levels of tricalcium phosphate. We expected to see a halo around the bacteria to indicate that the mineral phosphate was being taken up. We used wild-type Rhizobium tropici with an empty plasmid as a negative control, so no results were expected. We used Pseudomonas fluorescens as a positive control, as literature indicated it had phosphate uptake abilities. However, no halo was visible for any of the samples, providing our experiment did not work as planned. We believe this is due to the media we use, as the literature from which we derived the experiment used different media. For future experiments, we are trying these plate assays again with the media used in the literature to see if we can produce similar results. To test compatibility of different plants with Rhizobia, we grew and inoculated 4 species of plants in African violet soil. We picked 4 types of plants, 2 that are known to make root nodules with Rhizobium, the common bean and soybean, and 2 plants that are native to the eastern U.S., showy pink trefoil and American senna. Once a week, we measured the plant's growth and looked for nodules. We only observed nodulation in one of the showy pink trefoil plants, so we did not consider these experiments a success. However, this is the first time our species of Rhizobium has been recorded having a symbiotic relationship with the trefoil. After discussing with an expert, Dr. Sharon Long, we changed the temperature of the plant's environment from 20 degrees Celsius to 30 degrees Celsius and began measuring the pH of the soil to see if it was affecting the plant's growth, and we are waiting for these results. Overall, we were able to introduce our genes into Rhizobium and have initial evidence that at least one protein is expressed. We also have made good progress in finding the right conditions to test for phosphate uptake and for nodule formation.
As a diverse high school team, we understand that many young scientists do not see themselves represented in STEM fields. To address these feelings, we talked to educators about what they needed to emphasize diverse representation in STEM in the classroom. They validated our concern that diversity in STEM subjects is currently under-prioritized, and they provided inspiration for the materials we created, for example, by suggesting that we provide supplemental videos for classroom use. Our goal was to reach and inspire students, and we found the best way to reach them was by developing a series of posts on Instagram. The Diverse Science series highlighted a different scientist each month, relating to the diversity, education, and inclusion theme of the month. In addition, to reach those in traditional classroom settings, we created a calendar featuring one scientist each month with information about their background and research. In producing this calendar, our goal was to create an easy and accessible way for science teachers to be able to provide role models and information on the groundbreaking research that these scientists are accomplishing. We hope this will motivate more diverse and informed students to pursue their interests in the field of STEM. This year, we are addressing 3 Sustainable Development Goals. The first is Goal 11, Sustainable Cities and Communities. Our project is focused on remediating phosphate pollution in areas where fertilizer runoff damages the waterways and in turn disrupts communities. We discussed our project with the Baltimore representative Robin Lewis, and she affirmed our project's relevance to increasing the sustainability of our communities. The second goal is Goal 12, Responsible Consumption and Production. Since phosphate reserves are being depleted, phosphate is not only a dangerous pollutant, but a waning resource. If farmers were to use our Rhizobium construct, it would make phosphate more available to the crops, eliminating the need for fertilizer, which is decreasing in availability. This contributes to a circular economy, as well as responsible reuse of resources. The third goal is Goal 14, Life Below Water. Our project is the bioremediation of a pollutant that creates dead zones in waterways, so our project is promoting clean water. We chose to meet with stakeholders that had different perspectives on our project's relationship with these sustainable development goals: farmers, regulators, nonprofits, and scientists. We contacted scientists in the field, advocacy associations, proposed end users, and regulatory organizations to ensure that our work is responsible and good for the world. We contacted Dr. Anand Rao, Dr. Uda Pashovsky, Dr. Dave Jackson, and Dr. Reed Longley during the engineering design process. From learning more about selecting our plant species to working with the mechanisms of phosphate mobilization, their advice has helped us to ensure that our project idea is a valuable contribution and a worthy use of Rhizobium. The Chesapeake Bay Foundation, a nonprofit that advocates for the bay's health, discussed the role of public and pollution issues, the ethical concerns of our project, and our proposed implementation plan with us. We altered our implementation plan to close systems in ditch runoff banks based on their suggestions. We interviewed Claggett Farm, and they helped us to understand the importance of taking economic sustainability into account when marketing our project to farmers. Additionally, we reached out to Pivot Bio because of their experience in working on designing bacteria in order to provide farmers with more efficient and sustainable nitrogen to fuel their crops. Their expertise in navigating the regulatory and approval process has been valuable as we work on applying our proposed solution to phosphate pollution. Lastly, we contacted the U.S. Department of Agriculture to receive advice regarding the safety of implementing our project. Our contacts at the USDA guided us through the requirements and restrictions for permit applications for our project and showed us how to address any ethical or safety concerns. Our team believes that this is a project worth exploring beyond iGEM. The aforementioned vision will inspire us to take the next steps to develop partnerships with stakeholders so that we can pursue implementation beyond iGEM. This could entail further research, practice runs in closed environments and then in the field, developing a business plan for implementation, or even partnering with an organization with the resources to complete the project in full. Thanks to the improved abilities of our modified Rhizobium strains, this bacteria could become the future of combating nutrient runoff and improving the quality of our waterways. Thank you.
Rise Up, tackling the root of the issue.